With this afternoon's session. It's my great pleasure to introduce Tyler Jacks, who will be our second keynote of the meeting. I think many of you know Tyler quite well. He's been really a thought leader in cancer biology for the last 25 years, which is remarkable since he just turned 30. So, and I think many of us have used the mouse models that Tyler's produced, but he's perhaps been the most effective at using them, most recently focusing on areas of sort of cancer heterogeneity and cancer immunology. And so we're looking forward to hearing his work this afternoon. Thanks, Tyler.
Thanks very much, Scott. Some of those early thoughts were formed by Scott Lowe himself back many years ago. Let me thank the organizers for the invitation. It's a pleasure to be here. It's always fun to come to Cold Spring Harbor. I also want to apologize to the organizers and to the attendees that I arrived late and I'll be leaving early, so I apologize. I will be around shortly after my talk in case there are burning questions or issues. So what I decided to talk about today was tumor evolution, and the title suggests that we're going to do so in a newish way by doing it at single-cell resolution. And I'm actually going to describe 3 different methodologies that we are using in my lab to try to understand the course of tumorigenesis through the lens of individual cells. Here are my disclosures. None of them directly affect the work that I'll present today. So by way of framing, we know that cancer cells arise from normal cells through the acquisition of mutations in oncogenes and tumor suppressor genes, and increasingly we know that that's not the full story, that what I call here epigenetic changes likewise contribute to the development of tumorigenesis. Many of the emergent properties that cancer cells acquire that they need for successful tumorigenesis occur through epigenetic alterations and changes in gene expression. We also know that cancers don't arise alone. They do so in concert with other cells, the cells of the host, which influence tumorigenesis profoundly, and that's been a major topic of this conference. Fibroblasts, other stromal cells, and certainly immune cells shape the development of a tumor, and those interactions are extremely important. Because cancer is a dynamic process, takes place over time, and cells change over time, and because cancer cells interact with cells of the normal host in that process, many years ago, we decided that it was a wise choice to study the disease in situ in an animal model where we could access the tissue of the tumor and its surrounds at will and at different time points. So we began working on genetically engineered mouse models of cancer, and we've worked on many of them over the years. Today's talk will focus exclusively on one of those models, which has been referenced in this meeting, the so-called KP model of lung adenocarcinoma. It actually has its origins in the hands of David Tuvison, now at Cold Spring Harbor, who made the first KRAS stop-lox allele, which is featured in this mouse. This mouse also has a mutation in the p53 tumor suppressor gene. Both of them are CRISPR regulatable, so that you infect the animals with a virus that carries Cre recombinase. RAS becomes active, p53 is lost, and that's enough to initiate tumorigenesis. And then tumors develop from there, from individual normal cells. And the normal counterpart is the alveolar type 2 cell, one of the cells of the alveolus, along with alveolar type 1 cells. Just within a few weeks, one can observe histopathology, hyperplasias. These give way to adenomas, which give way to local adenocarcinomas that can be locally invasive and eventually metastatic disease. So you can see the whole range of tumorigenesis unfold in this model over the course of weeks and months. And that's an attraction to us because we want to understand what underlies these transitions. We really want to understand what events take place, whether they be genetic or epigenetic or what have you, that allows these transitions to occur and underlies the different biologies that occur in these different stages of tumorigenesis. Now, the first and most obvious explanation for how these transitions occur, especially following the sort of golden era of cancer genetics, is that following the mutation of KRAS and p53, and I should have said these are hallmark mutations in human lung adenocarcinoma, that other mutations would occur. Somatically acquired mutations in other genes would occur, which drive tumorigenesis forward. And as such, we were surprised to learn that that's not true in this model. When David McFadden and Katie Paletti, along with Greg Hannon, Harold Varmus, and others, teamed up to sequence the tumors of KP lung adenocarcinoma, they were surprised to find that there are very few protein-altering mutations. On average, there's 3 to 5 protein-altering mutations per tumor, and very few, if any of these, are recurrent mutations. So you cannot make the argument that tumorigenesis is driven by somatically acquired mutations. Now, there are copy number alterations that take place during tumorigenesis, and in particular, KRAS oncogene gets amplified in the transition from adenoma to adenocarcinoma, and there are a few other frequent or recurrent copy number changes, so those likely contribute at some level, but we don't believe those are the driver events. At the same time, we do know that epigenetic alterations take place, and we've known this for a while too. Monty Winslow, several years ago in the lab, was studying the transition of tumors from primary to metastasis and did bulk RNA sequencing, and we saw clear changes in gene expression in those lesions during progression. And in particular, for example, Monty found that a transcription factor called NKX2.1, which is a very important lung lineage-specific transcription factor, very much required for lung cell identity, went from being highly expressed in early lesions to underexpressed or not expressed in late-stage lesions. You can see that here. NKX2.1 very strongly expressed early, becomes spottily expressed in mid-grain tumors, and in late-stage tumors is gone. That's not due to mutation, that's due to an epigenetic event. It leads to the silencing of that gene. I like to think of it, it unlocks the cell from its fate of being a lung epithelial cell, allowing it to adopt perhaps other fates. Subsequent work by Eric Snyder found that in addition to losing lung cell identity, during tumor progression, the cells actually acquire alternative identities. They begin to express features of gastric epithelial cells, and they're marked by a transcription factor called HNF4 alpha, which you think about mostly in the gut and in the liver. That might surprise you until you realize that the lung actually derives from the gut in embryogenesis. So as these tumor cells become less lung-like, they become more familiar to the cells that gave rise to them, and that may be important as they begin to develop towards a malignant cancer. Moreover, Monty showed that another transcriptional regulator, HMG-A2, became upregulated in late-stage cancers. This is not normally expressed in the lung at all. It's restricted to embryogenesis, but it comes ectopically expressed in late-stage cancers. And you can see that here. Here's a grade 3 cancer, which has a NKX staining region and an HMG-A2 staining region. This is intratumoral heterogeneity. The cancer cells are changing. And the question is how? What underlies those transitions? Along the way, Tuomas Tammela, now at Memorial Sloan Kettering, made another interesting observation related to intratumoral heterogeneity, the observation that stem-like cells arose, actually, particularly in this transition from the adenoma stage to the adenocarcinoma stage. These cells were marked by being active for Wnt pathway signaling. And even more interestingly, Tuomas found that in the same tumor right next door, there were Wnt-producing cells, so that the tumor actually recreated a stem cell niche-like relationship, where there were Wnt-responding cells, shown here in green, and Wnt-producing cells, shown here in red, marked by an enzyme called porcupine, which is required for the post-translational modification of the Wnts prior to their secretion. So the tumor was actually recapitulating a stem cell niche-like behavior. And by the way, both of these cell types, Tuomas believed, and we now know is true, were cancer cell derived. These are not fibroblasts infiltrating in to create a niche for the cancer cells. These are cancer cells adopting different fates that were presumably selected for during a successful tumorigenesis. So that's a little background to set the stage for what we've now done, and everything from here on forward is unpublished data. These experiments largely relied on information we already had, as well as some bulk RNA sequencing that Monty Winslow initially did. But over the last few years, as you know, and speakers have discussed, we have the ability to do RNA sequencing at the single cell level. And we decided this would be a great system in which to do it, because we could look not just at a time point or at a late-stage tumor, but we could look at tumor evolution. We could sample cells at different time points and look at their transcriptomes, not in the bulk, where you lose information because of averaging, but at the individual cell level. And we teamed up with Aviv Regev's lab directly across the street from us at the Broad Institute, who had developed much of the technology that we needed, as well as the bioinformatic analysis. And I want to be clear now that this work was started by Tuomas Tammela in my lab. Tuomas now has continued the project in his own lab at Sloan Kettering. And Nemo Marjanovic, who's a joint student between my lab and Aviv's lab, was a principal on the project, along with David Canner. And Matan Hafri was the bioinformatician in Aviv's lab, who did all of the bioinformatic analysis that I'll tell you about today. So here's the setup. We isolate cells from either normal alveolar type 2 cells, and these are cells that are marked by a red fluorescent protein, and we use a CRI that activates that protein directly in type 2 cells. We also used KRAS-only mutant mice, as well as KP mutant mice that I've described to you already. They also have tomato, and we isolate cells at early time points, these stages, mid time points, and late time points. All the cells are marked by tomato, so we can isolate those cells. So all the profile I'm going to show you now is cancer cells. There's lots of interesting cells in a tumor. We're not talking about those, just the cancer cells. So we've isolated 4,000 or so cells at these different time points. And we've used SMART-seq2 as opposed to 10x sequencing. That's because that's what was available when we started. And it's somewhat advantageous because you actually get more genes per cell by this method than you do with 10x. So you actually dig a little deeper into the data by this technology. So what do you get? You get a lot of cells on a two-dimensional plot like shown here. And this is a plot that probably everybody's become familiar with, the so-called T-SNE plot, which is a dimension reduction algorithm that allows you to take very high dimension data and show it in 2 dimensions. And roughly speaking, cells that are close to one another in this plot have a similar transcriptional profile, and cells that are further apart have a less similar transcriptional profile. That's about all you can say. And you can't also learn very much from what I'm showing you here because all the identities of the cells has been grayed out. So you don't know who's who here. But if I reveal for you who's who, it looks a little bit more interesting. And here I'm using a color coding scheme that's recapitulated here. So the normal cells are green, and the early stage cells are blue, and the later stage cells are orange and red. And maybe you can begin to appreciate that things look quite interesting. As the tumors develop over time, they become less lung-like. They begin to change in their differentiation state, their gene expression state, their transcriptional state, as we progress through time and through stage. And you also might be able to see that there's certain structure that exists at these later stages. It doesn't seem to be random. Cells are collecting in transcriptional states, which is interesting. Now that's a little bit hard to look at, so I've broken it down by time point here. So we're going to play the movie of tumorigenesis, as it were, and hopefully this will work. So I'm going to unfold the different time points. So as you can see, as we move from normalcy to full malignancy, there's a northeastern migration. And it's not just that. There's structure here, too. There's clusters. If you look at these late time points, the 30-week time points, these are adenocarcinomas. There's clusters of cells. Now, you might say to yourself, that's actually not that interesting because they isolated a bunch of different tumors, and maybe those cells in those clusters simply reflect cells from different individual tumors that took different paths to becoming tumors, and thus they have different transcriptional states. That's not true. If we look at those 30-week time point cells, they do indeed line up in these different clusters. But if I now apply the identity of those different tumors, because we know which tumor is which, we find that for most of those clusters, there's representation for most of the tumors. It's not chaos. The cells have gone down common transcriptional paths towards these late stages. So that's interesting. That suggests there's some underlying biology that they're being selected for. It's guiding their development. So what else can we learn? Well, we started by looking at things we knew about already. Where are the genes that we talked about before expressed within this continuum? For example, I mentioned NKX2.1, which is the driver of alveolar type 2 cell identity. And one of the targets of NKX2.1 is surfactant protein C. And you can see that surfactant protein C is expressed very heavily in normal alveolar type 2 cells and in the early stage tumors, but at later stages, its expression gets lost, just as we would have thought. Here's HNF4 alpha. It's not expressed in the early stages. It becomes ectopically expressed as the cells begin to change towards that gastric phenotype, and then actually goes off again. And here's HMGA2, more restricted and specifically expressed in a subset of the most advanced cells. I also told you about Wnt, and indeed we can see signatures of Wnt signaling as shown here. A subset of the cells show Wnt positivity as based on a gene expression signature of Wnt signaling. And likewise, porcupine, that enzyme that modifies Wnt, is expressed in a subset of cells, mostly in the mid to late stages of tumorigenesis. We also looked at the more advanced stages of cancer. These tumors will eventually develop towards metastasis, and they undergo an epithelial to mesenchymal transition, the EMT, and we can see evidence for that. In this particular case, just this little tiny cluster over here, which is marked by expression of ZEB2, one of the EMT markers. So we can see the various stages, some of which at least we knew about. But we also learned things we hadn't expected. And in this case, probably we should have expected it. Here I'm showing another two-dimensional dimension reduction algorithm called fate mapping, and I'm switching now because the analysis from this point forward involves fate mapping as opposed to t-SNE. You can see that with this algorithm, the same general patterns are seen, early to middle to late. The structure looks a little different. That's not too surprising. Fate mapping, like UMAP mapping, actually gives you better understanding of the relationship between clusters that are close to each other, which is not really true for t-SNE mapping. But you can see the basic structure does exist here. And we then started to look at these using gene expression signatures of various things that existed in the literature. And we found one interesting observation, and that is that in that middle zone, what we would call cluster 3 in this analysis, I guess I haven't introduced that yet, there was strong expression for a mixed lineage, alveolar type 1 and type 2 cells. Alveolar type 2 cells had changed their identity towards alveolar type 1 cells. There was a form of lineage infidelity, as Elaine Fuchs would call it, where the cells are getting confused as to what they are. They're still lung epithelial, but they're in the wrong lineage. And that's actually not hugely surprising, because in fact, alveolar type 2 cells share a progenitor with alveolar type 1 cells. And alveolar type 2 cells are known to give rise to alveolar type 1 cells, especially during injury. So they have the capacity to, it's called transdifferentiate here, but some people like Mark Krasnow think that there's a subset of type 2 cells that are actually stem cells that give rise to type 1 cells. So the fact that we're seeing this transition from type 2 to type 1 in tumorigenesis maybe isn't hugely surprising. And I think Scott would agree with this. This might look like injury repair. And my guess is it is. And this is just kind of a natural reaction of these cells to the tissue disruption that's taking place within a developing tumor. So this is the fate map again, and here I have put the cluster names on. That's cluster 3 I was just talking about. Our attention got drawn to cluster 5 here. Cluster 5 became interesting to us for a variety of reasons that I'll come to. Here it is in isolation. These cells up here share a transcriptional program and thus are clustered together. So we call that cluster 5. And one of the genes that's in cluster 5 is SLC4A11, a gene you certainly haven't heard of, I hadn't, and probably has nothing to do, interesting to do, with the biology of what I'm about to tell you. It's just a marker. It's a marker of the cells in cluster 5. Okay? It's a borate transporter, for God's sake. So why is that cluster interesting? Well, the reason it's interesting is that it's present quite consistently across late-stage tumors, and that's shown here. Here are late-stage lung adenocarcinomas, and you can see that cluster 5 in blue is well represented in the single-cell data from all of those tumors. But note that it's also present in early-stage lung adenocarcinomas, as well as in adenomas. And so it comes up early, and it hangs around. And that was interesting to us. We also checked to see that it was not just the protein, RNA level, but there were protein equivalents in developing tumors. And so we used SLC4A11 antibodies, and you can see that there are, in fact, positive cells in grade 3 and grade 4 tumors, and occasionally also in grade 2 tumors. And by the way, this isn't just true of this mouse model. The group then looked in a limited data set from human single-cell RNA-seq, published by others, and indeed, in those two, one can find cluster 5, well represented, actually, in both studies. So that was exciting. But what was more exciting was when Matan used algorithms to look for the relationship between how these clusters form. You might be familiar with terms like pseudo-time analysis or optimal transport. These are methods, bioinformatic methods, that allow you to look at a data set like this and say, who came from whom? What is the transit map that explains how one cell became another cell and gave rise to another cell? You can do that bioinformatically by basically looking at the gene expression overlaps between sets of cells and inferring where they came from. And frankly speaking, that's about all I know about it. But these methods, and the method they used was called Waddington optimal transport, allow you to draw these transport maps. They kind of look like the subway system of New York City, or worse. They look like that. There's lots of things going in and lots of things coming out, and each of these numbers actually reflect these clusters here. And you might notice that cluster 5, the one I've been telling you about, sits right at the middle. It's the grand central station of this process. Lots of things come in, lots of things go out. You can actually see that a little more clearly if you break it down and just focus on cluster 5, and the purple lines reflect clusters that are thought to move into cluster 5, and the black lines clusters that are thought to emerge from cluster 5. So there's a lot coming in and a lot going out. And that stands in very stark contrast to cluster 11 down here. That's the EMT cluster, by the way, where nothing comes out of there. You only get to there. That's a terminal destination. Okay? So cluster 5 seems to be interesting because it may be a highly plastic state. The gene expression program there reflects a cell that can move in different directions. And you could imagine that might be an interesting thing in the course of tumorigenesis. So Tuomas and colleagues have now tried to explore that further, specifically looking at the plasticity component. And one experiment they've done is to isolate those cluster 5 cells from tumors using markers that they express on their surface, isolate those cells, take the rest of the tumor that doesn't have the cluster 5 cells, those are the high plasticity state depleted pool. Here's the cluster 5 cells. And then they also took the cluster 11 cells, these guys here. They call that a fixed state because it wasn't very interesting. They took those as single cells, formed them into organoid-like or tumor spheroid-like colonies in vitro, and then expanded them in vitro over the course of 11 days. If the high plasticity cells were plastic, their transcriptional program should change over time, whereas the fixed cluster perhaps wouldn't change very much. That was the experiment. And so they then did RNA sequencing on the cells that came out of that experiment here, and in fact, the high plasticity cell state showed the greatest degree of increased transcriptional expression or heterogeneity upon culture in vitro. And the fixed state showed very little. You can actually quantify that as shown here. This metric is an indicator of the degree of transcriptional homogeneity. The higher the number, the more homogeneous, less diversity. These cells had least diversity. The depleted pool had some, but less than the high plasticity state that had the most. They've also tested the biology of these cells. They've tested their ability to form tumor spheroids in vitro, and the high plasticity cells have a greater capacity to do that compared to the bulk cells of the tumor. An indication that they might be more tumorigenic or more malignant. The actual tumorigenesis assay is ongoing now, and we don't have the answer to that yet. If you transplant these cells back into an animal, are they more capable of forming a secondary tumor, as would be predicted? Moreover, they've taken these data and asked, all the data, but we'll focus on cluster 5, and asked, if you look at these clusters and the gene expression signatures that reflect these clusters, all of them, and compare them against TCGA data, do any of the clusters correlate with better or worse prognosis in humans? And the answer is, yeah, one does correlate very much with poor prognosis in humans, and that's that one, which is cluster 5. It's actually worse prognosis than cluster 11, which is the EMT cluster. So there seems to be some correlate to progression and prognosis in human cancers as well. So that's the single-cell gene expression data. We've learned a lot by studying that, and it's opened up lots of new questions to ask. And among those questions is, how do these gene expression programs come to be? What's driving these altered transcriptional states? What are the transcription factors? What are the epigenetic regulators that underlie these transitions? So the next story I'd like to tell you is our efforts to answer those questions. And this is a project in the hands of Lindsay Lefebvre, postdoc in my lab. And she's worked very closely with Jason Buonrostro, whose lab at the time we started was at the Broad Institute and is now at Harvard College. And Jason was actually one of the people who developed attack sequencing, chromatin profiling, when he was a graduate student with Will Greenleaf. And so he knew a lot about that, and we were very attracted to using attack sequencing as a way to understand the epigenetic or chromatin profiles of these tumors, again, through their evolution. So Lindsay teamed up with Jason to use, initially the thought was bulk attack sequencing on these tumors, but right at that time, Jay Shandori and others were developing single-cell attack sequencing. So we decided that we would try that. And Lindsay and Jason and their team perfected the method of single-cell attack sequencing using what's called split pool sequencing. And I won't go through the details of that, but suffice it to say, it's a method that allows one to get attack sequencing data from individual cells. And you can understand why that's valuable because, once again, you're not averaging information across populations of cells. And the method works really well, actually. Here's an example from their data where a bulk tumor sample is shown here. Here's the attack-seq peaks. Every peak indicates more open chromatin, open accessible to transposition, which is what's indicated here. So the higher the peak, the more accessible. You can see nice peaks here. And this is actually the aggregated data from a whole bunch, 12,000 single cells. And you can see that they're virtually identical. So the single cell data is quite robust, actually. So what did she do? Well, she mostly focused on samples from late-stage tumors, or animals with late-stage tumors. She allowed animals, these KPT mice, to develop tumors to late stage, and then she isolated late-stage tumor-bearing lungs and did attack sequencing on everything, or rather on the tomato-positive cells from those samples. She also did isolate some late-stage tumors, and she also isolated some cells from lymph node metastases, and those will become interesting. Now, you might think to yourself, why do it that way? Why wait till late stage? Why not do it like they did the single-cell stuff? Why don't they just take cells at different stages and do attack sequencing of them? And that's actually ongoing now. This is how we started. And I will remind you that there's a lot of heterogeneity, even at the late stage. And to emphasize that point, here's an example of heterogeneity. This is a 35-week tumor-bearing lungs from a single animal. And this is computer-assisted histopathology. We now have an artificial intelligence-driven model that allows us to automatically do histopathology on these samples, made by a company called Aphoria with help from Peter Westcott and Tuomas Tamala in the lab. It automatically calls grade 1, grade 2, grade 3, and grade 4. And you can see that you've got lots of grades represented, even at the late stage. Some grade 1 stuff, a lot of grade 2, mostly grade 3, and some grade 4. And you can also see intratumoral heterogeneity, like this tumor here that's got a lot of grade 3, but some grade 2, and this tumor here that's got a lot of grade 3 and some grade 4. So there's both inter and intratumoral heterogeneity represented at this late stage sample, so we thought there'd be a lot of richness to learn from there. So what do you get? You get a blob. This is the blob that ate Cold Spring Harbor. This is 12,000 plus tumor cells and 130 cells derived from lymph node metastases, all lined by t-SNE, and here the data input is the size of those peaks, not the level of gene expression, like on single-cell RNA-seq, but the size of those peaks across the genome. So you get a blob. The blob by itself doesn't look that interesting, but we believe the blob is quite informative. It's actually teaching us about tumor progression. And I say that because we have certain landmarks that let me say that tumor progression actually proceeds from left to right across a continuum. And I say that in part because we've actually put some markers down. We've profiled normal lung. So we know what the chromatin profile looks like in normal alveolar type 2 cells. And they're right here at the left edge, somewhere in San Francisco. We also have alveolar type 1-like cells. Those are actually alveolar type 1 cells sitting on top of some cancer cells. Now we've grown to expect that. We knew that there was a transition towards this state, and those cells sit here. So we believe that tumor progression moves from here, the cell of origin, in this direction, in part because here's where the metastatic cells live. They're all clustered right there, actually. And note, some tumor cells, cells derived from primary tumors, live right there too. So we believe that tumor progression moves from left to right. Again, you might say, well, how do they know that? They did have a bunch of different tumors, and they even had some isolated tumors. Maybe these different regions in this plot just reflect the outgrowth of individual tumors with distinct states, like I talked about before. But that's not true. If we look at two individual tumors and ask where do the cells from those individual tumors fall on this plot, they fall across the continuum. There are some, quote, earlier stage cells and some later stage cells. And that's true of all the tumors that we looked at. So they all represent cells along this continuum. Moreover, we're confident that it goes from left to right because we've looked at some of our familiar friends, like NKX2.1. We've asked the question, if you look at all the NKX2.1 predicted motifs in the genome and ask, do they change in an interesting way across this continuum? Not just NKX2.1 gene expression, but all the peaks associated with NKX2.1 binding. You find that in the left edge, those peaks are open, and in the right, they're closed. NKX2.1 went from being active and functional to being inactive and not functional. And you can see an example. Here's SPC. Here are two NKX2.1 peaks, accessible peaks in the early stage lesions, not accessible in the late stage lesions, exactly as we would predict. I told you about HNF4 alpha. Not relevant early, becomes relevant middle. And here it is. HNF4 alpha accessible peaks present in this middle zone primarily. Not so much early, not so much late. And we talked about EMT, a late marker. And sure enough, it's actually only found, or largely only found, in the cells in the metastasis or those tumor cells that look like the cells from the metastasis. I also told you about AT1, the fact that there's transdifferentiation, lineage infidelity during tumor progression is evidence from the single cell data. And sure enough, we can see that too. Here's a module of chromatin profile that we associate with AT1-like cells. And sure enough, those cells all have that module. And again, I remind you that AT2 cells can give rise to AT1 cells through this process. So many of the things that we've seen from these data support this transition, this set of transitions that we had seen from the single-cell RNA-seq data. But these data also give us insights that we had not expected. In a completely unbiased analysis of the peak changes and modules that were changing across the chromatin profile, Lindsay, Jason, and colleagues discovered that a transcription factor we had no understanding was important in progression was changing in interesting ways, and that's RUNX2, part of the RUNX transcription factor family. It went from being hardly involved to be highly involved, and this is actually RUNX2 attack-seq data. It's not on in early stage lesions, and it becomes open and active in late stage lesions and drives the expression of a series of RUNX2 target genes we had not appreciated before. On in late stage, on in those very late stage and metastatic cells as well, and that overlaps nicely with the twist data that I showed you before. So RUNX2 becomes interesting to us. Perhaps it's a driver of the transition to late stage progression. Perhaps it's a driver to the transition to metastasis. And I'll come back to that point in a second. We also focused a bit on these cells, which were quite remarkable, actually. The fact that all of the metastatic cells lined up so well together. Now, I remind you there's 130 of them, so it's not a huge number. And they were all derived from 2 lymph node metastases, not from metastases from all over the body. But they all lined up right there. And importantly, as I hope you might be able to see, certain of these gray cells were there too, and those were derived from primary tumors. And that was quite interesting to us. And in fact, it was not an isolated case. Cells that had that profile were prevalent in metastases, 100% of the cells, as I said. But if you look across several tumors, you find cells that have the exact same profile. And the suggestion from that is that those are cells that gave rise to those metastases. Those are the pre-metastatic cells. Those are the metastatic cells of origin. And they had acquired an epigenetic state that favored metastasis. That's a hypothesis. There's another hypothesis, and that is that metastatic cells somewhere circled back and reseeded the primary tumor. We actually can't exclude that. But the fact that it's quite prevalent makes us think it's probably not true, because some of these animals with these tumors did not have evident metastases. That's not proof, but that's at least a suggestion. Moreover, I just remind you that these tumors are heterogeneous. like this tumor here, which is one I showed you already. It's got regions of grade 4-ness, as evidenced by aphoria and HMG-A2 staining. And we believe that these cells are arising from those lesions, or those portions of lesions, although we haven't proven that point. So I want to come back to RUNX2. As I said, it was the important new discovery enabled by the attack sequencing. And RUNX2, you may know, is not known to be important in lung biology at all, actually. It's known as an important bone development factor, important in osteoblast differentiation. It's actually co-opted in cancer, in certain hematopoietic malignancies and in breast cancer, and certain other RUNX family members likewise. But it was not known at the time to be important in lung. So we began to look at it, explore it functionally. And what Lindsay has done is to modify RUNX2 expression using CRISPR in cell lines derived from our model, originally made by Monty Winslow, actually. Cell lines that are described as metastatic, that is, they have metastatic capability. And these cells at the outset actually express high levels of RUNX2 as shown here. And we also have cell lines derived from the model that are non-metastatic, and their levels of expression of RUNX2 are lower. And then we use CRISPR to modify RUNX2 in these cell lines, to knock it out, for example, or to overexpress it using CRISPRa, to test the idea that RUNX2 activity could affect the chromatin state of these cells, as well as their biology as it relates to metastasis. And in fact, by modifying RUNX2 in these ways, you do affect the chromatin state. Here are chromatin accessibility changes in the knockout cells and the overexpressed cells, and they're actually inverse. We find a bunch of things that become accessible when we overexpress RUNX2, and these very same things become reduced in their accessibility when you knock out RUNX2. And very interestingly to us, many of these genes encode factors that are implicated in the extracellular matrix, secreted factors of the extracellular matrix, which may be relevant to bone biology, but also cancer biology. Maybe the RUNX2 expression pattern is actually creating a cell which is influencing its extracellular matrix, tumor microenvironment, in a way that allows those tumor cells to progress more rapidly to metastasis. And in favor of that idea, Lindsay has recently done some tail vein injection studies looking at metastatic capability of these cells, in which she's knocked out RUNX2 compared to the control metastatic cell line. And indeed, when you knock out RUNX2 compared to the control where there's lots of lung metastases in this tail vein injection assay, knocking out RUNX2 essentially eliminates that. So we believe that RUNX2 probably is guiding the development of this pre-metastatic state, which is then maintained in the metastatic condition. In the future, and actually ongoing, we're modifying the developing cancer cells with CRISPR to modify RUNX2 in both directions to see if we can test that in vivo. And finally, in this section, we, like we did with the gene expression data, have asked whether the attack sequencing data can teach us something about human lung cancer. And in fact, we've observed that a module associated with the late stages of progression from the mouse data actually does nicely predict poor prognosis in humans, and that's shown here. This set of genes, which are identified by attack sequencing from the mouse, actually very nicely separates those with advanced and aggressive disease from those with less aggressive disease. All right, so I'm going to end. I realize I'm a little bit over. Are you guys okay with that? By coming back to a question that I raised earlier, and I did it specifically in the context of the observation that there were cancer cells in the primary tumors that had a profile, epigenetic profile, similar to the metastatic cells. And there were two possibilities. Either one gave rise to the other, or the other gave rise to the first, right? And you can't easily distinguish those two things from what I've showed you so far, but there is a way to do it, and that's using lineage tracing. And so what I'd like to tell you about now is an ongoing and relatively early stage project, which is being carried out by Dian Yang, who's actually in the audience, who's a postdoc with Jonathan Weissman, and is collaborating with Santiago Naranjo in my lab to use CRISPR-based lineage tracing to understand clonal development in this same model. There's a lot of details here, and we can go through it later if you're interested. But this is a technology developed in Jonathan's lab to allow one to follow cells over time based on the acquisition of CRISPR-induced mutations into target sites that you've engineered into the cell. And in our case, we've engineered 30 target sites for CRISPR to modify. And the hope is that CRISPR will do that over time as the tumors are developing and create scars in those target sites that allow us to figure out which cells descended from which, because that pattern will be maintained by cell division. And one can actually create very detailed maps of lineage development, and this has been applied very nicely in Jonathan's lab to real development, but here we want to apply it to tumor development. And so with Santiago and Britt Rideout in my lab, Dion created embryonic stem cells that carried these 30 CRISPR target sites along with the sgRNAs that target them. These cells had the K and P mutations as well. We then took these embryonic stem cells following Scott Lowe's playbook and made chimeric mice that are partially composed of those cells, and then infected those animals with a lentivirus that carries CRI in order to activate RAS, inactivate P53, and activate Cas9, and then let CRISPR do its thing. Hopefully not all at once, but over time, to create a lineage map of the cells that evolve. And I should say that this is read out not at the level of DNA. This is actually our expressed targets, and you read out the information at the level of RNA. And that's quite useful because you're going to capture RNA from these cells, and therefore you can capture not just the target site RNA, but also the rest of the RNA, and ultimately figure out what the gene expression pattern of that cell was. So does this work? The answer is remarkably yes. Here's a tumor, a single tumor, and here are the various sites. As I mentioned, there's 10 integrations with 3 sites each, 30 sites, and the colors indicate the fact that a cut has occurred there, a discernible cut has occurred there, and if it's a different color, it's a different cut. And based on this information, you can actually recreate the lineage history of each cell in the tumor. And we're looking at now hundreds of cells within a given tumor. And you can make certain predictions about clonal evolution depending on how you think things actually unfold. It could be that there's actually no selection, that every tumor cell is equivalent, and the descendants of every tumor cell will contribute to the eventual tumor that you see. You would call that neutral evolution, and you would see a very complex pattern as shown here. It's also possible, and I think we would imagine it the case, that as the tumor develops, certain cells have acquired certain properties that are advantageous, and that clone will take over. That will be clonal expansion or clonal evolution, and then you would see the outgrowth of a dominant clone eventually. So we actually, so far in its early days, see a little bit of both. In an early-ish stage tumor, 20 weeks post-initiation, we can see examples like that, where the clonal architecture of the tumor is quite rich. This looks like neutral evolution. And other cases like this, where we see a lot of richness, but then a dominant subclone starting to emerge. All of these cells reflect a cell that had that as its common ancestor. So that's outgrowth of a clone. And then at later stages, even less complexity, where this tumor probably derives from 2 dominant subclones. So clonal evolution. Now in the future, we're going to be able to use that not just to understand the clonal evolution of a primary tumor, but we can also isolate metastases from these same mice and figure out where did those metastases come from? What gave rise to what? Moreover, as I mentioned, there's going to be gene expression data, and it's already coming, from those very same cancer cells. So we'll know their lineage, and we'll also know their gene expression. So we'll be able to prove the point that there's plasticity within the gene expression state, that there's overlap between clonal evolution and epigenetic changes that I talked about before. We're not there yet, but we'll be there soon. So I'm going to finish there and just remind you that I focused entirely on the cancer cells. I didn't talk about any other cell type, but we all know that they're really important too. And you can use these exact same methods to look at them. And we're beginning to do that on the T cell compartment in these tumors, but you might think about doing the exact same thing with fibroblasts and macrophages and so forth. Not at one time point, but dynamically as these cells change over time. So I want to thank many people who've contributed to this work. As you can tell, it's been a lot of work. The single-cell RNA sequencing project was initiated by Tuomas Tamala, Nimo Mranovic, as well as David Kanner, in close collaboration with Aviv Regev's lab, and is being continued in Tuomas's lab. Lindsay Lefebvre, collaborating with Jason Buonrostro and members of his lab, developed the single-cell attack sequencing method, and all the data that I shared with you today. And the lineage tracing project is a collaboration between my lab and Santiago Naranjo and Dian Yang, who has really pioneered the use of this technology from Jonathan's lab for this purpose. And I'll stop there and thank you for your attention.
Excellent. Questions?
Thanks. This is a very beautiful study. So my question is, because your tumor cells are RTD labeled, so I would think it'll be easy to identify those circulating or disseminated tumor cells and then to see where they fit into the big picture, because those will be very important transitional phase that bridge the primary to metastasis.
You're asking an excellent question, which is, what does the profile look like in the circulation? And because they're fluorescently labeled, we can isolate them. And we're starting to do that. I don't have an answer for you, but it's a great question. And we actually have a collaboration. The inventor of the technology is sitting in the second row, not just to exsanguinate a mouse and isolate circulating tumor cells, but to plug a mouse into a CTC device, which allows us to take cells longitudinally from the same animal. So that'd be a terrific application of exactly what you're talking about. Yeah.
All right.
Thanks very much. This was fabulous. So when you're looking at the tumors as they develop, you showed, grade 4 in the center of the tumor. Is that because that region is becoming more hypoxic and these cells are adapting to the hypoxic stress?
We don't know the answer to that, but it's certainly possible. It's not the case that all grade 4 regions are in the middle, but I'm sure that hypoxia is part of the story. And that allows me to make another point, which I failed to make, is that looking at things in by imaging will give us a lot more information still. It's not something we've put into the studies to date, but some sort of spatial analysis will be also very important to overlay with the gene expression data and so forth.
You have seen cases where a clone can, there is a bias to a particular clone and it takes over.
Yeah.
Is there any evidence that the cells generating these clones are from cluster 5, the plastic cluster?
That's a great question. Dion and I actually had lunch together today. He works in San Francisco, so he only visits occasionally. So I talked to him about that very thing. And I think the answer is we don't know yet, but that's the next step. We're starting to get gene expression data from those same studies. So the question is, where does that signature live? Can you see evidence that that signature drives that kind of expansion? Or can you see evidence for that signature being represented in a subset of the cells that look like that, which would reinforce the notion that it actually is a highly plastic state? We don't know yet. Somebody's speaking. Yeah.
My turn?
Where are we? Back here. Here we go.
So for cell in cluster 5, when it's highly plastic, what are their surface marker and can that tell you anything about what they are?
Yeah, it's too early to say, and it's frankly now transitioned to the work of Tuomas's lab. So there are markers that allow us to isolate those cells, but we're not talking about them now.
Yeah, so I was wondering if the original genetic lesions in the KP model trigger just the initial events, and then it's like sort of a cascade downhill, or whether there's continuing functions of the RAS and so on that are required throughout progression.
Well, I think we know it's.
You probably don't know the answer to that.
So I think that obviously it's there and important at the beginning, but I think we know already that it's also important throughout. Scott's done some beautiful work on just targeting by drug inhibition the pathway, and even late-stage tumors remain sensitive to inhibition. You have to hit them pretty hard, but you can do it. P53 likewise. Loss of P53 actually probably isn't that important very early in this process, but in that middle zone, we know from work that we've done and others too, if you flip P53 back on in those middle stage tumors, make it active, you get a very strong response. So I think that it's not just limited to tumor initiation.
Very beautiful study. So I was curious with one topic, which is you showed between mice the expression clusters more or less remain the same.
Yeah.
So I'm wondering about within a mouse, is there intra-tumor heterogeneity for these phenotypic gene expression clusters like we think about from mutation heterogeneity we see in human cancers?
You're asking, is there a difference between within an animal and between animals?
Within animal,
Yeah.
is there intra-tumor heterogeneity for existence of these gene expression clusters? Do cells exist in different phenotypic states within a tumor?
Within a tumor, yes, and between tumors, even tumors of the same animal, yes, and between tumors of different animals, yes. So all are
of
true.
them.
So Tyler, one thing that puzzled me, the data was really clear, is that the so-called high plasticity cell fate was an indicator of particularly bad prognosis
Yeah.
in human
Yeah.
patients, but not the EMT cluster.
I know.
So
Well,
what are your
it's
thoughts?
not quite true that the EMT cluster wasn't. The EMT cluster was actually second in the list
see.
of good clusters. So it does predict poor prognosis, just not as well as this cluster does. So that's the argument. This cluster is especially interesting. But the EMT cluster is bad too.
I see.
Back here.
Any correlation between RUNX2 and seeding to bones?
Great question. We've never found metastasis to the bone in this model. Having said that, we haven't looked that hard. So now we're starting to look harder.
In the clinic?
Well, in the clinic, it's not known. We do know that, and I didn't say this, but RUNX2 actually itself is correlated with poor prognosis. Whether it's correlated with bone metastasis per se, I don't know the answer to that.
I have a following question. So interested in the RUNX2, the finding of RUNX2 in that metastasis clusters. So you have definitely several genes there.
Yes.
So knockout RUNX2 will ablate this
Right.
metastasis, sufficient
Right.
and necessary. Do you expect that other individual genes also
Yeah,
sufficient and
it's a
necessary?
good question. You're absolutely right. It's not the only gene in that cluster. We think it's a major driver of that pattern that we observe. So it may be a master regulator of the downstream things that you observe, and maybe that's why we see the strength of the phenotype that we do. But it doesn't preclude the possibility that there are other genes that are contributing, and if you did functional studies on them, you'd likewise see an effect. So the answer is possibly yes. There's more to find there, but RUNX2 is the only thing that we've demonstrated functionally so far.
Thank you.
So I'll ask it maybe oh, no, there'll be more. So I'll ask them. The lineage tracing stuff is very cool. Not surprisingly, some of your other progeny have thought about playing around with that too.
I know.
But in your case, when you see the parallel evolution, and maybe I'm just not thinking about it clearly, couldn't that just be multi-clonal tumor initiation? In other words, that they're just different.
Sorry, there's a detail that I didn't mention. Dion would have corrected me if I had forgotten this. Part of the setup is that it's a lentiviral CRI, and the virus integrates and expresses its own barcode.
Okay.
So you actually can tell one tumor from another.
Got it.
So these are all derived from the
That's very
same
cool.
cell of origin.
Very cool. Okay, back here.
Yes, going back to your cluster 5, do you know and can you share whether it has partial EMT features or cancer stem cell features?
It has some stem cell features, yes. It has some, I wanted the Wnt pathway signature to be so strong there, but it's actually not so, it's distributed in and around cluster 5. But there are other things that look STEMmy in that cluster. If you looked at a gene expression signature of stemness defined by others, you'd find it fairly enriched in that cluster. So yes, there's evidence of a stem-like profile within cluster 5.
And no partial EMT? Sorry.
I'm not trying to be evasive. I simply actually don't remember whether there is or there isn't.
Okay, last one, Stella.
A little bit following up on Scott's question concerning the tumor tracing.
Yeah.
Do you have an experimental system to kind of uncouple tracing induction from tumor generation, that you could basically toggle on and off Cas9 to get kind of dynamic tracing at different stages of tumor formation?
It's a great question. And so Dion has a future plan that includes everything under the sun, including that. And you definitely can modify the tracer in lots of different interesting ways. Like you said, turn it on and off with Cas9 being regulated, right? You could make Cas9, or the guides, but let's just say Cas9, be regulated by specific conditions. Hypoxia. When a tumor enters a hypoxic state, what happens to clonal evolution? When it enters, you know, whatever condition, what happens? So you can make it conditional in lots of different interesting ways. So I think that will be even more exciting for the future, yeah.
All right, great. Thank you, Tyler. That was fantastic.